Hi Academy kids and Dr. Weber. This is Julie Burton, and for my individual class presentation, I was assigned Chapter 25, Section 4, which is called Evolutionary Developmental Biology. Now, before I begin my lecture, I want to go over my wiki page with you so you know where to go. Okay, this is my section summary, and the bold words are defined at the bottom here, quick vocab review, and it has all of those vocab words. Then my PowerPoint presentation, and I made this PowerPoint presentation, and it has all the same information as my Prezi, and it's just a straightforward way to read over the information and to study. My Prezi, this is what I'll be presenting within class. My useful materials, which I have not posted all of them yet, but they will be right there, and the exam questions, which the students can't see yet. Okay. This is my Prezi. I'll start my presentation. So my topic was evolutionary developmental biology by me. Now, what is evolutionary developmental biology? It compares the development of different organisms in an attempt to understand ancestral relationships between organisms and the developmental mechanisms that bring about evolutionary change. And this field of biology consists of the exploration of genes that control development in organisms. Now, these genes are called developmental genes. It's pretty straightforward. The nickname, which is much easier to pronounce, is devo, and that's how I'll be referring to it for the rest of the presentation. And it's a fairly new field of biology. The history. Researchers over the past few decades have become more interested in the similarities and differences between closely and distantly related species. Now, as I stated before, developmental genes, and they're very important to the phenotype of an organism, and they're key players in the evolution of many traits. Sorry about that. Changes in genes affect traits that can be acted on by natural selection. And also, variation in the expression of these genes may be commonly involved in getting new traits. Next, pattern formation, which is the process that gives rise to plants and animals with a particular structure, and developmental genes control it. Now, first, a real-life example. The morphological differences between webbed and not webbed feet are due to different expressions of two cell signaling proteins. These proteins are called BMP4 and gremlin. Now, what is BMP4? BMP4 is expressed throughout the developing limb, and it causes the cells to undergo apoptosis, which is programmed cell death. And gremlin, on the other hand, inhibits BMP4 from working, and it allows the cells to live. Now, for our scenario. In a chicken, gremlin is expressed throughout the development of the limb except in between each digit. So that means that since the gremlin protein is the one that inhibits the BMP4, which causes apoptosis, the gremlin is expressed throughout the development of the limb except in between the digit, which means that the gremlin does not stop the BMP4 from killing the cells in between the digits of the foot of the chicken. So that would result in not webbed feet. Now in the duck, gremlin is expressed everywhere, which means that the gremlin throughout the leg stops BMP4 from killing the cells, and that causes webbed feet. This is a picture of the chicken and the duck, and clearly the chicken is on the left and the duck is on the right. Now and then, you can't assume that scientists would just leave it at that. They have tried inserting gremlin into the interdigit regions of the chicken into the interdigit regions of the chicken, and it was born with webbed feet. And this is that chicken, and as you can see, it has webbed feet. Now, on to our next topic, Hox genes. Now, the definition of Hox genes is that in animals, it's a class of genes involved in pattern formation in early embryos, and Hox genes is short for homeobox-containing genes. And they have been very important in observing trends among large groups of species. They're found in all animals, and that shows that they originated very early on in animal evolution. Now, scientists believe that a variation in Hox genes may have started the formation of many new body plans. Sorry about that. Next, the number of Hox genes varies per animal. Now, to explain this further, this is a picture from the textbook, and you see there are many different types of animals on the side, such as sponges, insects, and mammals. And the pink section, the pink strip, is the chromosome, and along the chromosome are strips of blue, purple, orange, and green. Now these multicolored strips on the pink chromosome are the Hox genes, and as you can see, sponges only have one Hox gene, which is blue, and well, in this picture it's blue. And flatworms have 4, and then when you get to mammals, there's an abundance of Hox genes. So as you can see, the number definitely varies per animal. Now, multiples occur in clusters. As you can see, there are multiples. Going back to this picture, there are multiples in the mammals. Now, they're close together on the chromosome, and in mammals specifically, Hox genes have duplicated twice so far to create 4 clusters, all somewhat different, and there are 48 genes in total. Now, this is pretty much an estimation. This is just a generalized number, and it's not necessarily true for every single mammal. And the more Hox genes you have, the more complex the organism. They are directly related. So, that goes to show that a sponge would definitely be less complex than a mammal because it has less, well, one of the reasons is because it has less Hox genes. Moving on. Developmental genes are affecting growth rate and influencing morphology. Now, controlling relative growth rates in different parts of the body during development will cause genetic variation, which makes sense. Now, heterochrony. The definition is evolutionary changes in the rate or timing of developmental events. Now, this can cause 2 things. The first is different species with morphological changes. An example is the human and chimpanzee skulls. The different parts of the skull grow at different times, making the skulls look very different from one another. Now, the second thing that can happen is it can affect development stage at which a species reproduces, which is very, very interesting because it can do 2 things. Either parts of the body associated with reproduction develop faster than the rest of the body, or reproduction could occur at the same known age, but non-reproductive parts would develop slower. Now, both will cause paidomorphosis, which brings us to our next real-life example. Brooke Greenberg. For background information, she was born January 8, 1993. She's currently 18 years old, and she lives in Baltimore, Maryland. Now, this is what she looks like today. Doesn't look right. But why? The reason is she's paidomorphic, which means the retention of juvenile traits in an adult organism. Brooke suffers from what doctors call Syndrome X, which means they don't exactly have a diagnosis for her. It's an absolute mystery to all scientists and doctors. And because she has the mental capacity of a 9-month-old and looks like a 13-month-old, but she's 18. Now I'm going to stop this to show you a video for just a few minutes about Brooke. Fast forwarding. Now what is going on now? Because they are looking at her genome and they're seeing that nothing is really wrong with it because it looks the same way that a normal, well, she was 16 at the time, so a normal 16-year-old girl's genome would look. So this is the huge mystery that's going on. And scientists are searching for gene mutations or any abnormalities in her DNA to see what is causing her not to age. Now we're thinking that if the scientists do find the aging gene, the aging gene, that she could be the ticket to the fountain of youth. And this is the end of my presentation. Thank you for listening. Make sure to read my section summary and review vocabulary and use my PowerPoint to study from. And the useful materials are very interesting, and you should be set for my section in the exam. Okay, thanks for listening.